Hello, Derek.
Hi, Eugenie.
So we are at Singularity for the second interview with Singularity students. So Derek is one of our brilliant students from this year from Canada. So Derek, I wanted to know a little bit more about your background.
Sure. I was originally a psychology major at Rice University in Houston, and then ended up going to Microsoft, doing user studies, user research, and software design, and then finally working for several years in Microsoft research on speech recognition. This came from a background in statistical software development, and so was working on the engine, but was also working on the user experience of speech recognition. Primarily speech recognition on mobile devices, so what you would want to say to your personal organizer, for instance, if it could understand everything that you said. And this was interesting from a human interaction standpoint, in that we were working on visions that still aren't fully captured, even though the personal organizers have gotten a whole lot better than they were in the days when I was first working on them. Unfortunately, though, Microsoft did not get better at the same rate the devices did. Microsoft started feeling somewhat confining in terms of just being a huge company, and it went from being a very entrepreneurial place to work to being somewhat more bureaucratic and locked up in a bigger company. And so I found to be always climbing the steep side of learning curves that I had to go elsewhere. And I'm actually now back at the University of Victoria in Victoria, British Columbia, and am primarily studying synthetic biology at this point.
Excellent. So what kind of project are you working on in synthetic biology? Could you describe them a little bit more?
Sure. So again, on climbing the learning curve and changing areas from computer science into synthetic biology, it was very important that I was able to develop some of the lab skills. And there's a competition that MIT sponsors every year called iGEM, or International Genetically Engineered Machines. And basically what happens in this competition is that you start with a library of parts. About 1,300 parts actually are given to you on three 384-well trays. And you can put these parts together in just a myriad of ways to create exciting novel new organisms. And even just the words, create exciting novel new organisms, are kind of futuristic and science fiction-ish. And so me and a group of other students started this iGEM team to compete in last year's competition, and we built a bacterial thermometer. So most of the parts are on E. coli, which many of your listeners might think of as this scary pathogen that makes you ill when you eat bad hamburger. But in fact, the lab version is very benign. You could drink a quart of it and not be terribly ill. And it's used because it's the research backbone for much of synthetic biology at this point. And so you take these parts together and assemble them, and so our part, when it was at normal room temperature, would slowly grow and just be an average bacteria that didn't do anything special. But when it got over 42 degrees, the bacteria would begin to glow green. And so it's a bacterial thermometer.
Excellent. And so now you are in our amazing school, Singularity, and you told me that you wanted to work more on startup in synthetic biology right after, and as well as the technology is ready. So what are we waiting for?
So I went back to school trying to decide whether I was going to go an academic route or go back into industry or go into a startup of some form. And the startup route is most appealing to me from many perspectives. It's just where things move fastest. You get to work on the most variety of things. And with synthetic biology and the computer science, I'm developing a very well-equipped bag of tricks and wondering where to take it. It's becoming the biggest hurdle is not the technology, but the regulatory aspects. And this is very frustrating in that bureaucracy and red tape and difficult to move organizations are why I left Microsoft and went back to school, quite bluntly. And so the side of biology that I'm most interested at this point in is the do-it-yourself biology movement, the small startups, and finding the spaces that are not under the sway of giant lobbying efforts, new regulatory efforts, being able to start something innovative and new.
Okay, thank you.